So we're now going to move into our next external guest speaker, Dr. Stroeher, who's going to talk on basically soil microbiology and its role in soil health. Yui graduated from the University of Adelaide in 1993 with a PhD in microbiology, where he remained for a further 6 years as a postdoctoral research scientist on aspects of cholera virulence. In 1998, he was awarded an Alexander von Humboldt Research Fellowship and subsequently spent 2 years working in Germany. On his return to the University of Adelaide, he worked on Streptococcus pneumoniae for 8 years before moving to the Hepatitis Research Laboratory. During that time, he started working for Nutrog as an independent consultant on the development of products such as go-go juice, which is commonly used, at least in garden horticulture. In 2010, Yui moved to Flinders University working on Staphylococcus aureus and Acinetobacter baumannii resistant antibiotic treatment. Since 2016, Yui's been working full-time as the R&D manager in Nutrog. Very much. Welcome you, Yui, and look forward to your presentation. Thank you very much.
Firstly, thank you very much for the opportunity to present here today. Look, I know that my background may seem like I'm all about pathogens. In many ways I am, but I'm a repressed ecologist, believe it or not. So being back in looking at soil ecology is actually pretty exciting for me. So just very quickly, Nutrog has been around for about 36 years. In the last 16 or so years, we've really focused on doing R&D in respect to soil microbiology. We've put together a biological advisory board which has some quite well-known and I guess respected scientists on it. So there's Professor Paul Manning, who spent a significant amount of time working at AstraZeneca. He just had a small budget of $500 million a year to spend, half his life. Associate Professor Renata Morona is an expert in a whole range of pathogens and has a very extensive background in microbiology. And lastly but not least is Professor Kirsty Bayless, who actually works on a whole range of fruits and nuts, including things like macadamias. May have some relevance here. So anyway, I'm really here to talk about biologicals and what they can do for you. So you may be using biologicals in your farm already, you may not be, but biologicals can really be split into three main groups. So we have what are known as biofertilizers. So that's really about making those nutrients that are locked up in your soil available for microbes to use, for plants to use. So we call that bioavailability. Bioavailability just means it's available for the biology of the plant. So you're looking at things like potassium, phosphate, iron, and various other cations. And also you have the ability to fix nitrogen. So I don't know if any of you guys are growing cover crops like clover and that type of thing, so you get a significant amount of nitrogen fixation there. So we actually look at the microbes that don't associate with a legume, so we look at the free living ones. The next section is what you would call the biostimulants. So they're the things that you might find in an extract of say seaweed or humates or even plant growth hormones directly. So you have things like amino acids, vitamins, humates, and maybe silicon. And then the last area is what we call the biopesticides. So the biopesticides are really about microbes that reduce the disease burden by killing or inhibiting pathogens in your soil. And that occurs via three mechanisms as a rule. The first is competition. The competition is simply to drive the pathogen out. So think about a healthy lawn. A healthy lawn doesn't allow any weeds to grow, therefore there's competition. It stops the pathogen from coming in this case the weed. There's contact killing, which is some fungi and some products work by contact, so trichoderma, if any of you using trichoderma inoculants, They tend to work by contact killing, by parasitosing other fungi. And then there's a way in which microbes secrete antimicrobial compounds. So think about things like antibiotics or antifungal agents. And so these are really the three main regions where biologicals play a role. Now, those biologicals can then be split into effectively two groups. Those that contain living organisms and those that are basically extracts, so non-living biologicals. And these are really the differences that you see. So think about if you inoculate the soil with a microbe, let's say it's a bacillus species, because that's a good example. There are lots of products out there with a bacillus. Serenade, for example, is an example. That bacillus will basically grow in your soil, and what it will do, it'll colonize maybe around the roots. It might drive out disease, and what it can do is it can continue to basically produce the products that it's using, or the biosynthetic compounds that are being produced by that microbe will continue to be produced in the soil. Whereas a non-living biological, whatever you put on, once it's used up, it's used up. There is no opportunity to get more. The other thing is they can colonize roots. They can react to changes in the environment. So if you think about a plant when it's stressed, it'll be sending quite different signals into a ground than when a plant that isn't stressed. So what you need is you need a biological or an inoculant that can actually respond to the plant's needs. So think about it, if a plant is being attacked by certain types of pathogens, for example, Pseudomonas syringes, which is a major pathogen, it'll actually send malic acid into the ground to recruit bacillus species, because bacillus species are natural biocontrol agents for some of these. So again, you get a longer term benefits, you get short term benefits, but both tend to give you a benefit. So for me, it's all about microbiology. So what are the main aspects? So you got nutrient acquisition. So I'll talk about breaking down organic matter, making nutrients available. So you might be putting out phosphates or potassium-based fertilizers. Are you getting the maximum use of those? Are they actually, are those nutrients actually available for plants to use? Increasing carbon dioxide availability. I'll talk a little bit about that. Probably more an issue for new plantings. Nutrient manufacturing, as I mentioned, that's things like nitrogen fixing, disease protection. I won't be talking about stimulating plant defences, and also the production of plant growth hormones. So let's start with the fungi that associate with your roots. So in your pistachios, there will be basically mycorrhizal fungi. For something like a pistachio, it'll be what we call an ectomycorrhizal, so it'll form a sheath around the root. But in essence, what you have is you have a root, and basically you have those root hairs, and you see that dotted line there, that's what we call a depletion zone. So within that zone is the area where the plant can have ready access to nutrients, okay? So unless you've got soluble nutrients like nitrates or sulfate, they can't move into this depletion zone. So if you put phosphate down here, it won't move across. Phosphate is insoluble, it doesn't travel through the soil. So what these fungi do is they form these hyphae, they interact with the roots, they form these hyphae, and now you end up with being able to access nutrients outside of what's known as the depletion zone. So significant increase in nutrient acquisition. So if you have a look at this little pine tree here, all that sort of fluffy material around here, that's basically all the fungi, so that's not actually the roots. Okay, so that's one thing that they do. The other thing that they do is they actually reduce your nematode burden. So hopefully this video will play. It did before. Come on. There we go. So you'll see these little nematodes wiggling around. These lines here that you see is basically a network of hyphae, so fungal mycelium in the soil. And so now what happens is these nematodes get stuck onto those hyphae. The fungi looks upon a nematode as a little bag of protein. Okay, so if you think about a nematode, it's mainly made out of protein. And what's protein? It's a source of nitrogen. So what these fungi do over a period of about 45 minutes, obviously this is a time-lapse video, I don't have 45 minutes to show the whole thing, but you can see by now the majority of those nematodes have been digested away. All right, so there's a lot of fungi that you can basically encourage in your soil in order to get nematode protection. However, if you go in with high levels of nitrogen fertilizer, so if you start throwing out the ammonium nitrate or the calcium nitrate or the urea, the fungi will be quite happy to stay in your soil, but they won't switch on this killing system. It's exactly the same reason I don't cook if Uber Eats comes and delivers to me every single day. Okay, I don't bother to put the energy in. So in order to extract that nitrogen for the fungi, they need to expend energy. If you're giving them all the nitrogen that they want and all the nitrogen they need, they'll switch that system off. So yes, use nitrogen fertilizers, but be aware that if you use too much, you'll switch that system off. Now, in respect to nitrogen fixation, so you're probably familiar with those nodules that you get on clovers and on peas and on beans. These little nodules is where nitrogen fixation occurs. So this is an example here. This is basically a peanut plant. This is one that hasn't been inoculated with a microbe that helps to fix nitrogen. This one has been, so you can see quite a significant difference. Now, why don't all plants basically form this association? That's because the plants and the microbe have to have basically a handshake, okay? In this example over here, what they've done is they've basically done exactly the same experiment, but they've knocked out one of those hands in the handshake, so they've knocked out a key or a lock. So although the microbe might be present, the plant no longer recognises it, and therefore doesn't form that association. You don't get the nodules, you don't get nitrogen fixation.
So what's the lock?
Well, the lock in this case is the rhizobium, so the bacteria secretes something called a nod factor, nodulation factor, and the plants produce a flavonoid, and only when those two come together does the plant recognise that microbe. So the nod factor gets recognised by the plant. If that's there, they'll send out flavonoids to recruit more of these microbes, and they'll form as an association. So for an association with a fungi, the fungi will send out what's called a MIC factor, so a mycorrhizal factor, and the plant will send out stringolactones, which is basically another compound. And when those two meet, again, you get that handshake, and the plant will then allow those bacteria or the fungi to form an association. Without that handshake, they basically can't meet up and marry, so to speak. Now, obviously, not everybody's going to be growing clover or beans or peas in their orchard, but there's a range of free-living microbes, and so there's quite a lot of those, and they'll fix about 30 to 40 kilos of nitrogen per hectare per year. Now that's not a huge amount, but it still equates to probably somewhere around 80 kilos of urea equivalent. So again, you want to encourage these types of microbes to be in your soil. Now, as I mentioned before, phosphate is basically a nutrient that gets locked up. So phosphate is negatively charged, and so anything in your soil that's positively charged will be attracted to it. Think about it like a magnet. So phosphate is negatively charged, calcium is positively charged. You get an attraction. So you get calcium phosphate. When you have calcium phosphate, that phosphate is not available for plants to use. They cannot take that up across their membrane. They can only take up free phosphate or free calcium. Similarly, if you've got iron in your soil, you'll get iron phosphate. So anything that's positively charged, magnesium, calcium, iron, aluminium will tie up phosphate. So what these microbes do is they uncouple that. So you can see around some of these microbes, you've got that clear zone. Don't know how well that comes out here. There's a clear zone around this microbe and that one, and not these ones. These guys here are unlocking phosphate. So they're basically taking that bond, they're releasing the calcium, they're releasing the phosphate. The plant now takes up the calcium, the cation, and the phosphate as the anion. Okay? So it's a way of unlocking phosphate that may be in your soil. So when you get a soil test done, you'll see total phosphate and you might see available phosphate. And you'll often see that available phosphate maybe only be 10% of your total phosphate. So how about we try to extract some of that phosphate that's locked up? Similarly, if you have potassium in your soil, lots of soils have potassium. Any soil that's rich in granite, mica, feldspar tends to have potassium locked up. Very similar thing here. You can see that cloudy area, that's insoluble potassium. Around these microbes here, you get that zone of clearing. That's available potassium for plants to use. And this is a very simple system. This simply involves these microbes secreting a small amount of organic acid. So when we're talking about organic acids, we're talking about things like lactic acid, citric acid, malic acid. So all of these acids help to dissolve basically these, in inverted commas, rocks or insoluble material to make them soluble for the plant to take up. Now, carbon dioxide is an interesting nutrient because a lot of people, are really worried about the level of carbon dioxide we have on the planet. And admittedly, increasing CO2 will lead to changes in our environment and increased temperatures. Currently, we're at about 400 parts per million. Now, at that level, you can see that some plants respond very positively to carbon dioxide, other plants don't respond to the same degree. So CO2 is known to be a fertilizer. So in countries like Iceland, what they'll actually do is they'll actually capture CO2 and they'll pump it back into greenhouses in order to increase the plant growth. So CO2 can be a limiting nutrient. So when you mulch or you slash your cover crop and you leave it there to break down, that releases CO2. Okay, so that actually can help your plants to grow if CO2 is limiting. CO2 may not be limiting, it may be another nutrient. If you want to know more about limiting nutrients, you can look up Liebig's law of minimums, and that basically explains how certain nutrient requirements need to be met in order to get full plant growth. So you may get an increase in plant growth, but the really interesting thing about CO2 is that when a plant is stressed for CO2, so when CO2 levels are very low, it's in my pocket, there's pores on the base of the leaves. So when the CO2 levels are low, the plant opens up those pores in order to get CO2 in, because the plant's looking for CO2 in order to photosynthesize. When CO2 levels are high, the plant closes those pores, okay? So when you have a big wide open pore, what happens? Water vapor leaves the leaf. And so basically, if you have a low level of CO2 around your plants are more likely to get stressed due to drought, okay? Because they're losing more water. High levels of CO2, smaller pores, less water loss. Therefore, your water use efficiency starts to go up as CO2 goes up. So there's a reason why you may want to use mulches or compost or leave the cut material on the ground in order to get a small amount of CO2 in your thing. The other things that microbes are really good at is producing plant growth hormones. And as a general rule, they produce 3 of the major plant growth hormones. They produce what are called auxins. So you may be using things like rooting powders or rooting gels for establishment of new trees. Basically, that's an auxin. Cytokinins and gibberellic acids function slightly differently. They're a different type of plant growth hormone. They're more involved in shoot development and other parts of development of the plant. So microbes produce these plant growth hormones. So if you think about that, if you are putting out a new planting, you want to get good establishment, you want to get rapid establishment of new plants. And the idea is you want to get that rapid establishment before a heat wave comes along. Well, if the plant's producing more lateral roots, more roots in general, it's going to be more resilient to a subsequent heat event. Or, as we've seen at the moment, potentially a frost event. Okay, so cytokinins and gibberellic acids work differently. They are more involved in the shape of the plant and also how many leaves it puts out and the internodal length, so where the leaves come out. So the other things that microbes do is they produce humic substances in your soil. So when microbes break stuff down and when they die, what's left? No, it's not that. What happens is basically you end up with humic material. So you may be using humic acids as a liquid in your orchard. I don't know if anybody is. Again, basically they're extracted from brown coal or from peat. But in effect, what you end up with humic acids, which are quite high molecular weight, so they're very large. Fulvic acids are much smaller. And they will, over time, travel through your soil profile. And you have insoluble humics. So they are the sort of thing that if you have a compost and the compost has been going for a few years and you've got that brown black mass that's left over, that's basically a whole lot of insoluble humic material. Now, why is that important? Well, number one, we know it holds an enormous amount of water. But the other thing is they actually stop the loss of your micronutrients through the soil. Okay, so what happens is these humic substances have small groups on them. I'm sorry about the chemistry here. Have small carboxyl groups. So basically, all that means they've got the ability to capture micronutrients. So think about a micronutrient that you put on. You may be putting on iron, or you might be putting on magnesium or manganese. As you water that material travels through the soil profile. What the humates do is they capture it. They capture and release, capture and release. And so instead of it travelling through the soil like this, it travels through the soil much more slowly. And in that period, it means that the nutrients that you've got hang around the plant's feeder zone or root zone where their feeder roots are for a longer period, so you get better use, okay? And it simply works very similar to the phosphate story about positive and negative attractions, except that this attraction here is quite weak. So those micronutrients are released and captured, released and captured, released and captured. When they're in a released form, the plants will take them up. When they're captured form, they're simply sitting in the soil around your roots. As soon as they release, the plant can capture them again. Okay? So, obviously I come from Nutrog, and this is really what we spend a lot of our research doing. So we look at all of those aspects. We look at nutrient acquisition, nitrogen fixing, we look at inhibiting plant pathogens, we look at auxin production, and then we go, if we make a happy, healthy plant, wouldn't that happy, healthy plant then produce more roots, secrete more sugars into the ground, recruit more microbes, and therefore capture more carbon? So can we use happy, healthy plants where the soil is microbiologically active to capture more carbon? And if you're into carbon farming, and some people may be, that may be a really, really important subsequent income stream. So what do? I spend a lot of time in the lab ordering around my minions, and I spend a little bit of time actually doing some lab work. But what happens is that we get soil samples in, and in essence what we do is we look for different types of microbes. So we take the soil sample, we dilute it, we put it onto a growth media, and here you can see different microbes, different colours, different shapes. Some can be quite hairy and furry. Some may show a zone of clearing. So here's one that basically solubilizes potassium. So we start to look for all the different functions. So we look for things like the ability to solubilize phosphate here. There's two bacteria here and a fungi. You see that clear zone or that clear zone around this and this species here. Or we look for the ability to liberate potassium. Or in some cases, we look for the ability to actually make iron available for plants. So what microbes do is they secrete compounds that capture iron, and what the plant has done, or the plants have learned, is they've actually learned to hijack the bacterial system of iron acquisition and use it for themselves. So these microbes that have that yellow zone around them have the ability to capture iron that the plants can then subsequently use. This microbe doesn't. And we look for microbes that have the ability to break down cellulose. And why is cellulose important? Well, cellulose is important because, again, it's involved in that whole CO2 and nutrient cycling. I'll skip over that one. So we probably spend most of our time, however, looking at how do we inhibit fungal diseases in plants. So at the moment, we've got about 60 different fungi. So when I'm talking about fungi, I'm talking about things like your fusariums and that sort of thing. Then you got your oomyosites. So there's things like your Pythium and your Phytophthora. And then we also have started looking at some bacterial pathogens, so bacterial canker and a few others. And so basically what we have all these different pathogens. And then what we do is we put those pathogens onto a media that allows them to grow. They'll grow in a radial pattern. Above and below, we spot what we think might stop them from growing. And so here you can see, basically, that's Pythium. It'll grow up this way, but it won't grow down here. Okay? Here's Fusarium. Again, same sort of thing. The Fusarium will grow everywhere, except for, where we put our inhibitors, and so on. Here is one we did for is an almond hull rot, Rhizopus. Same thing, we put the pathogen in the middle. You can see that zone of clearing around here. Here's Phytophthora. So Phytophthora canker in almonds. Again, the pathogen will grow everywhere. This one is inhibiting. This microbe up here doesn't. So we basically end up with a collection of different microbes. And so we were approached by Century Almonds, and I don't know, Gemma's not here. Ah, there. Sorry, I didn't see you. So we were approached by Century Almonds to see whether or not, well, or maybe we approached you guys. I can't remember which way around it went, but anyway. we were interested to see whether we could inhibit almond whole rot. And so initially we had a set of microbes that we work with, and we found about 6 isolates that had the ability to inhibit almond whole rot. We went back and we screened our bank, and our bank now consists of about 2,500 microbes. We screened about another 300 to 400 of those. We end up finding a whole range of other inhibitors. So we made a cocktail, and then in essence, that was sprayed into the canopy. And what you can see is basically that this biocontrol agent is significantly better than the current standard for inhibiting almond whole rot. And we've done this experiment for a second year and saw a very, very similar result. And hopefully, if Century Almonds are happy to keep working with us, we'll do it for a third year, just to make sure. So that's a biological control agent. It's a single application, and basically it probably, from a cost perspective, is cheaper than the current fungicide.
But these other ones also seem to have a significant
That's right.
effect.
So these are potassium
Potassium
silicates. Yep. You can certainly use those as well. Yeah, so I mean, it's all about what you want to use. So I mean, some of these potassium silicates are probably ACO certifiable or certified. This product certainly is ACO certified. So it also depends a little bit on what the grower requires and what he can do. But again, this is a single application, whereas the potassium silicate needed 2 applications. You see that level of protection. We've done something very similar in black dot in potatoes. Again, what we basically saw was about an 80% reduction in disease burden. Here, we took all the inhibitors that we had against black dot, formulated them together, sprayed them onto some pellets, put them in with seed potatoes. We did five separate areas, each of 20 hectares, and in effect we saw about an 80% reduction overall in the pathogen load on those potatoes that we treated with the biological control agent. So we ended up saying, well, if we got all these positive results, we should really be thinking about making a product that the growers can use. And so we ended up making a product called Populate. So Populate basically contains 42 specific microbes that we grow up in the lab. So each one is grown up separately, closed down separately, mixed together. And so what we did is we went through it and we said, we want to cover all the functions that I talked about: nutrient acquisition, disease protection, plant growth hormones. But we want to cover each of those functions multiple times. So it turns out that when you start looking at microbes, you see that lots of those microbes have multiple functions. So the idea is, let's take lots of different ones and put them together. And the analogy would be, I could take, say, the very best hunter in the Arctic, polar bear, and I could drop him into the Australian outback, and his chance of catching a kangaroo are probably pretty slim. But if I drop in a polar bear, a lion, a painted dog, a cheetah, and a leopard, one of them is going to catch a kangaroo, he's going to eat, he's going to survive, and he's going to multiply. The idea is diversity is really important, because we want to be able to have a product that will work here, or in Tasmania, or in Queensland. So at the moment, that product is Populate. Again, very similar, you see this range of inhibitions. I won't go through that again. I'll just give you another example. Here we used to Populate, again, pathogen loading on potatoes. Used at 5 litres a hectare, 100 days post-planting. Pathogen detection was done by Sardi in Predictor-B. You see quite a significant reduction in the pathogen loading. Another experiment we did with Century Almonds, or what Century Almonds did for us, was to basically look at Phytophthora in almonds, and you can see again significant reduction in the number of trees that were lost due to Phytophthora, with the exception here of one variety in one area. The numbers here are quite low, so that could be noise, but yeah. As a rule, 7 out of 8 plots showed some level of reduction of Phytophthora loss. This is a bit of data I stumbled across yesterday. Thanks, Gemma, for helping me out. Gemma sent me some data, and I got all excited because we're talking about almonds and completely forgot to look at the pistachio data that she sent me. Here, basically, the plots were treated at 10 litres per hectare, and you can see, in effect, that in all cases there was an improvement in the growth of the pistachios. Now this result is not statistically significant, this result here is. So what we're saying is that populate has a positive influence on a whole range of things. Now that may be disease protection, it may be plant growth, it may be nutrient acquisition, but our data to date shows that it seems to be working quite well in quite a number of settings. So as something completely different, let's get away from nuts for change. This is basically a field trial that was set up. This is a capsicum crop treated, untreated and treated, but the pudding's in the eating, as they say. This is basically what we ended up with the populate versus the untreated examples. Similarly here for tomatoes, significantly more tomatoes, but the interesting thing was that those tomatoes were actually firmer and had a longer shelf life than the untreated tomatoes. When we start looking at root development in tomatoes, this is a pot trial, significantly larger root mass in those that have been given populate versus those that were just given water. So we can show the root stimulating effect, we can show the disease suppression, we can also show obviously the increase in yield and quality of the food. If anyone here is growing any pasture crops, very similar result. The treated seed gave significantly more root mass and more nodule formation than the one that was treated with a trichoderma inoculum. In a nutshell, as they say, basically populate, you put it out at 5 litres a hectare. It can be applied to the foliage, but it's really designed as a soil application. It can be coated onto fertilisers. You can spray it, vertigate it. Generally we say 1 to 2 applications a year. So after extreme weather events, whether that's extreme waterlogging or extreme heat, we suggest reapplying. There isn't a withholding period, so all the strains that we work with have been sequenced, so we know exactly what strain we're working with. Every strain that's grown up is grown up in sterile media on site. So everything is done effectively like a pharmaceutical company. We don't drive around with a bubbler in the back of our ute with 2 shovels of cow manure and a bunch of molasses and tell people it's the greatest thing since sliced bread. At the moment, I'm working in a very small lab. This is going to be our new lab that's just begun the construction, or it's just about to start construction. This will be what's called a BC2 lab. A BC2 lab will then allow us to start bringing soil samples in from overseas. And also we want to then start looking at things like diseases we currently can't look at. I'll leave you at that, and I hope I haven't taken up too much time. I'm happy to field any questions.
Terrific, thank you very much, Yui.
That's all right. Thank you.
So questions around the soil microbiology?
What size containers does it come in?
5 litres, 20 litres, 200 litres and a shuttle.
Price per litre?
So price per litre, about, what's the price per litre? $12 roughly in a shuttle?
Yeah, about that.
Yep.
12 to 13.
Yeah, $12 to $13 a litre in a shuttle, yeah. 5.
Could this be described as the ultimate snake oil?
Well, you could argue that if you wanted to, but our data would show you that it isn't. I mean, we've done quite a significant number of trials now, and we've seen positive results. And, you know, we've just had a large order from Select Harvest. We've invested a significant amount to get it. Admittedly, you know, biologicals have had that snake oil
But some of the biologicals
work, yeah.
As said by Patrick Brown, who's a pretty big person, you know, get his name on a bit of paper.
So basically, yes, you could argue that a lot of biologicals are snake oil. We believe that by having that multiple combination of all the different microbes, that gives it a much better chance of working in the various conditions. Most biologicals that you'll find will have maybe 1 to 5 microbes in it. We have 42. So
Yeah,
there is some competition, but the main competition is actually from the ground you put it in. So we close them each down individually. So at that stage, they're in a state of stasis. So they're not competing each other at that stage. Once you put them out, the majority of the competition is actually due to the endemic microbes that are there and the selective pressure that's applied by the plants. So plants obviously also select those microbes they want to associate with, depending on the types of sugars they secrete. Now, most of the microbes that we end up with in our product are quite hardy, so a lot of them are spore plants, but others are delicate, what we call gram-negative bacteria. They are more delicate and they are more prone to drying, but as a rule, I wouldn't describe it as snake oil, but everybody has to make up their own mind what's snake oil, what isn't.
I'll just say that we put that populate on an area that was pretty badly affected, 2 to 3 year old trees with Phytophthora, and we noticed something straight away. I said, hang on, there's something going on here that we didn't know, and I'll put my hand on my heart. If you've got phyto problems, put it on, you'll see results. So it can't have been fluke, and we set that program.
How about storage and longevity? Is there use by date and what sort of temperatures?
So ideally, I say to people if they can use it within 1 year to 2 years, that's the use by date we recommend. Storage, as long as you don't store it in direct sunlight, try to keep the temperature below about 45 to 50 degrees. The microbes are pretty good. If you put it out in the bright sun or store it in a shed where the temperature is 70, 80 degrees, then obviously that's going to kill quite a few of those microbes in the product. Not all of them. Some of them are spore formers, as I mentioned, and you can literally almost cook the product, but you want to preserve as much diversity as possible. Yep.
Can you leach it out of the ground or leach out over irrigate?
The idea is that the microbes actually will, over time, or they will start to attach to organic material on the ground, in the soil. So they've got basically molecules that attach it. I mean, you really have to be flooding it. It's not about leaching them out. The only way that they tend to disappear is if you get a waterlogging system, where effectively what happens is the soil becomes anaerobic, no oxygen, so all those that are strict aerobic microbes will die in that system. So that's my timer. Time to get up. Make sure I didn't go over time.
So is there a particular time of day when they should be applied
So
to minimise
because
stability?
Yeah, because they're a living microbe, and I think somebody mentioned it last time, was that ideally out of direct UV light. So we suggest applying late afternoon or early evening, and ideally water it in. If you can get it watered in at least in the first, say, 4 or 5 mils of the soil, that means it's out of the UV. UV is the biggest killer. So if you think about in the old days, people used to hang up sickness in the house, everybody washed their sheets, hung it straight in the sunlight, because sunlight has got so much UV in it, it pretty much kills everything. Same with these microbes. So you try to avoid that. I mean, high UV is a killer, and really dry conditions are a killer. But I mean, all of you guys basically water anyway, so you irrigate. So moisture probably isn't a problem, but high UV can be. Again, if you put it out when there's foliage cover, or you put it out in the evening, or there's a rain event or overcast, then generally it's okay.
So what sort of filter would take out half your microbes?
So this is filtered down to 128 microns, is what we filter it to. As a rule, individual microbes, if you're looking at bacteria, they're probably somewhere in the vicinity of 1 to 4 microns, as a rule. Fungi tend to be bigger. The fungal spores are significantly bigger. So unless you're filtering down to, say, 20 microns or below, pretty much everything will survive that. Yep.
So this be distributed through fertigation?
Can be, yep. You just put it in through fertigation.
Fertigation.
Yep. Which is obviously the ideal way if you want to keep it out of UV light.
Yep.
Yep.
Was the number chosen after reading the Hitchhiker's Guide to the Galaxy?
No, but that's a good point, because 42 is the answer to everything. You are correct. Now, the number was chosen because what we did is we started off with a bank of microbes, and we initially had 50. And then what we did is we sent all of those off to get the genome sequence done. And when we got some of the data back, we realised that, well, obviously the soil contains good guys and bad guys. And so we had to exclude some from our list, and we ended up with 42, because some of the ones that we sent off didn't do anything, and others turned out to be really, really good at doing stuff, but turned out to be pathogens, which isn't really ideal for, either for humans or for your plants. So that's why we end up with 42. No, not the Hitchhiker's Guide to the Galaxy, but It's actually a good pickup.
What could a grower do to try to promote the natural indigenous microbial flora in their own soil?
Yeah. So I guess the most important thing above all is organic material. Organic matter is really the food source for microbes. That old adage that compost feeds the soil and doesn't feed the plant is very true. So basically, if you want to encourage a biologically active soil, you need to use compost or organic mulch, which will break down over time. The only thing with organic mulch is obviously you can get nitrogen drawdown if you use mulches that are very low in nitrogen and too high in carbon. But as a rule, yeah, organic matter is the key. Yeah. But remembering that 90% of your organic matter disappears back into the atmosphere, so you have to reapply it on a regular basis.
So would this be a lot better than getting a few truckloads of neutral fertilisers, as in compost and...
Well, the thing is that neutral fertilisers or compost are actually a fertiliser. So you're actually getting NPK, sulphur, calcium, all the rest of it. This is a liquid product that has virtually no nutrients in it. So it is basically a probiotic for your soil. So if you think about it, it's a bit like, how do I say this without dissing somebody? It's a bit like when you go and put out your humic acids, and sometimes you go, well, there's 26% humic acid, I'm putting out at 5 litres a hectare. How much humates am I getting? Probably not a whole lot, right? But nevertheless, people see a result, and it's the same with this. You do see a result. I mean, this contains the microbes, but it also contains a seaweed extract, it contains some humates, it contains some tricontinol. So there's some plant growth hormones in there, plus some other bits and pieces. So it's on a base, it's on an organic base, but it's not a fertiliser per se. It's basically a probiotic. Yes?
Spraying on the trees, because the pistachio is such a waxy leaf, is it going to penetrate through the leaf?
So yeah, so we've done some trials recently, and this is data I've only just got back, is with, let me just get this right. This escapes me now, give me a second. Myrtle rust in lemon myrtles in New South Wales. And yes, ideally, if you wanted to do that, you may have to use some sort of surfactant.
I missed that.
You'd have to use a surfactant with it. Yeah, if you had a really waxy leaf. Yeah. Any other questions? I mean, there are some very good surfactants out there you can use at quite low levels. Some of the organosilicates you can use at 1 in 2,000 dilution, in which case you're really putting in half a litre per 2,000 litres, or a litre per 2,000 litres. All right.
Thanks so much, Julie.
Thank you.
Really